The discovery of King Richard III's remains has completely changed what we thought we knew about his story and his bloodline. For centuries, his final resting place was a mystery. Historical records said he died at the Battle of Bosworth in 1485, the last great clash of the Wars of the Roses. Accounts claimed his body was taken to Leicester and buried at the Grey Friars Friary, but when the friary was later destroyed, its exact location was lost. Some even believed his body was thrown into a river. That mystery was finally solved in 2012, when archaeologists began digging under a council car park in Leicester, a place believed to be where the friary once stood. No one truly expected much to be found, but within days a skeleton was uncovered in the exact area where records suggested Richard III had been buried. At first, it was simply called Skeleton 1. But the clues it carried were extraordinary. The bones belonged to a man in his early 30s, Richard's age at death. The skeleton showed multiple battle wounds, including deep cuts to the skull, matching reports that Richard was killed brutally after his helmet was removed in battle. One of the most striking features was the curved spine. The skeleton showed severe scoliosis, which would have caused uneven shoulders. This detail matched descriptions of Richard from his own time, though Shakespeare later exaggerated it into a grotesque hunchback. In truth, the condition was real but not as deformed as legend made it seem. Other evidence supported the claim. Carbon dating placed the bones firmly in the late 1400s. Isotope tests from the teeth showed a diet rich in meat and fish, the kind eaten by someone of noble status. And most importantly, the burial site itself was exactly where the friary choir once stood, believed to be the king's final resting place. All of this pointed strongly to Richard III, but it was not enough on its own. Scientists needed absolute proof, and that meant turning to DNA. Modern genetics allowed something that would have been impossible in earlier centuries. Comparing the DNA in 500-year-old bones to living descendants. Richard III had no direct heirs, as his only legitimate son died young. But his family tree could still be traced. Researchers followed the maternal line, identifying descendants of Richard's sister and of York. Because mitochondrial DNA passes unchanged from mother to child, these living relatives could provide a match. At the same time, scientists studied the paternal line through the Y chromosome, passed from father to son. By comparing it with descendants of the wider Plantagenet family, they hoped to confirm Richard's identity as a true Plantagenet king. The results were astonishing. The mitochondrial DNA matched perfectly with descendants of his sister, proving beyond doubt that the skeleton was Richard III. But the Y chromosome told a different story. It did not match the living male line descendants of the Plantagenet family. This suggested a break in the royal bloodline, meaning at some point in history, a child was fathered by someone outside the official line. This revelation has left historians shocked and sparked fierce debate about what it means for royal succession and the legitimacy of certain dynasties. The discovery of Richard III's bones was already one of the most extraordinary archaeological finds in British history. But the DNA results have added another layer of mystery and controversy. Was Richard really the rightful king by blood, or does this discovery show that royal lineage is not as pure as once believed? What is clear is that science has reshaped our understanding of history, raising new questions about power, identity, and the secrets that can lie hidden in the bones of the past. The archaeological discovery in Leicester gave a strong case that the bones were Richard III's, but the world wanted absolute proof. Could DNA taken from bones buried for more than 500 years confirm the identity of a king? It was a difficult challenge. Ancient DNA is fragile, often broken down into tiny fragments and easily contaminated by modern handling. But if it worked, it could provide evidence stronger than any historical record. Scientists turn to the two kinds of DNA that remain almost unchanged through generations: mitochondrial DNA and the Y chromosome. Mitochondrial DNA, or mtDNA, passes from mother to child, unaffected by the father's genes. That means all descendants along the female line share the same mtDNA. Richard III had no direct children, but through his sister Anne of York, genealogists traced two living people who carried that same maternal line. Michael, a furniture maker from London, and Wendy, a genealogist in Canada. If the mtDNA from the skeleton matched theirs, it would strongly support the claim that the bones were Richard's. The paternal line was tested through the Y chromosome, which is passed almost unchanged from father to son. In theory, Richard III's Y chromosome should match that of other male descendants of his ancestor Edward III. Scientists turned to the Somerset family, descended from Henry Somerset, the 5th Duke of Beaufort, as they represented a surviving male branch of the Plantagenets. If their Y chromosomes matched the skeletons, it would be undeniable proof of Richard's identity and lineage. The process of extracting DNA from the remains was painstaking. In specialized laboratories, scientists worked under strict conditions to avoid contamination. Wearing protective suits, they drilled into the teeth and dense sections of bone, where DNA was most likely to survive. The fragments were carefully cleaned and amplified millions of times so sequencing machines could read them. Even then, the fragments were so small that researchers had to piece them together like a shredded manuscript, checking and rechecking to ensure accuracy. When the mtDNA results came back, the evidence was crystal clear. The skeleton's mtDNA matched both Michael and Wendy, direct maternal descendants of Richard III's sister. The chances of such a match happening by coincidence were astronomically small. This was powerful confirmation that the bones truly belonged to Richard III. But then came the twist. The Y chromosome results did not match. The skeleton's Y chromosome was different from that of the Somerset family and other Plantagenet male line descendants. This meant that somewhere between Edward III and Richard III, or even beyond, there had been what scientists call a false paternity event. In simpler terms, it meant that at some point in history, a child born into the royal family was not fathered by the man recorded in the official lineage. The child was accepted as legitimate, but biologically the bloodline was broken. This revelation sent shockwaves through historians and the public. Where had the break occurred? Was it early in the generations after Edward III, when questions of legitimacy already influenced the Wars of the Roses? Or could it have been closer to Richard III himself? Without more DNA samples from other royals, it was impossible to know for sure. What was clear was that the idea of a perfectly unbroken royal bloodline was more myth than fact. For scientists, the mtDNA match was enough to confirm the skeleton's identity as Richard III. But for the public and historians, the Y chromosome discovery opened a whole new mystery. It suggested that the Plantagenet line was not as secure as once believed, raising questions about legitimacy, inheritance, and dynastic claims. Instead of closing the story, DNA had written a surprising new chapter in royal history. The discovery of Richard III's remains opened up more questions than anyone expected. The DNA evidence pointed to something shocking. Somewhere in the royal family tree, there had been a break in the paternal bloodline. Scientists called it a false paternity event, but in simpler terms, it meant that at some point, a child born into the royal family was not actually fathered by the man listed in the records. The implications of this were huge. If the event happened early, perhaps among the sons or grandsons of Edward III, then the legitimacy of entire branches of the Plantagenet dynasty could be thrown into doubt. That would mean Richard III was not the only king affected. Many monarchs who followed might not have had the true bloodline at all. But if the break came later, perhaps in the Somerset line from which the tested descendants came, then the issue might not involve Richard's branch at all. Without knowing when or where the event happened, the mystery remained unsolved. This discovery carried enormous symbolic weight. For centuries, the idea of an unbroken royal bloodline was treated as sacred. But now science showed it wasn't as pure as people once believed. In fact, studies of modern populations suggest that false paternity events happen at a rate of around 1 to 2% per generation. Over many centuries, it would actually be unusual if a long dynasty avoided such events altogether. Still, finding evidence of one in the line of England's kings was startling. The public reaction was immediate. Newspapers speculated about medieval affairs, royal scandals, and betrayals hidden in castles. People wondered if this could have been the trace of a queen's infidelity or a secret relationship involving a noblewoman. Historians urged caution, reminding everyone that there was no way to assign blame or tie it to a specific event. The truth was both exciting and frustrating. The evidence of a secret was clear, but the details were beyond reach. For scholars of the Wars of the Roses, the irony was striking. Richard III's reign was consumed by arguments about legitimacy, with York and Lancaster each claiming the crown through their version of the family tree. Now, centuries later, DNA revealed that the entire idea of a pure royal line may never have been true. The wars may have been fought over claims built on shaky biological ground. Meanwhile, the skeleton itself continued to tell Richard's story. His scoliosis explained the uneven shoulders that contemporaries described, though it was far less severe than Shakespeare's portrayal of a deformed villain. His bones showed clear evidence of his violent death. Multiple cuts to the skull, likely from swords after his helmet was removed, and sharp wounds to the pelvis, suggesting humiliation after death. These findings matched written accounts that he fought bravely but was mocked once he was killed. A facial reconstruction added even more to the picture. Using CT scans, experts rebuilt his features from the skull. The result was a surprisingly human face, one that resembled portraits painted soon after his death. Combined with DNA evidence suggesting he likely had blue eyes and light brown hair, the reconstruction challenged the dark, monstrous image shaped by Shakespeare. Instead, it showed a determined man, not unlike the depictions preserved in the National Portrait Gallery. Other scientific evidence added to the case. Isotopic analysis of his teeth and bones revealed details about his diet and lifestyle. His childhood diet had been modest, but as he rose in rank, his adult diet became rich in freshwater fish, expensive meats, imported delicacies, and large amounts of wine. This shift matched what we know of Richard's rise to power, from noble to king. Together, the evidence built a powerful case. The mitochondrial DNA tied him directly to his family. The isotopes confirmed a royal lifestyle. The skeleton's injuries matched historical accounts. The face reconstruction matched contemporary portraits. Everything pointed to the same conclusion. The bones found beneath a Leicester car park were truly those of Richard III. Yet, the Y chromosome mystery refused to go away. It left behind a haunting question. Was England's royal history built on a bloodline that was never as unbroken as it seemed? DNA analysis added even more depth to the portrait of Richard III. The results suggested that he probably had blue eyes and light brown hair, features that matched early paintings of him. Later portraits, which showed him with darker features, were likely altered to match the image of a villain. The skeleton also helped separate fact from fiction. Shakespeare's famous portrayal of Richard as a monstrous hunchback was far from accurate. The spine showed that he did suffer from severe scoliosis, which caused his right shoulder to sit higher than his left and compressed one side of his ribs. This condition would have been visible, but it did not make him the deformed figure from the plays. With specially made armor and clothing, Richard could still move, fight, and function as a capable warrior. The battle wounds on his skeleton made that clear. He fought until the very end, even after losing his helmet. Deep cuts to his skull showed he had been attacked by multiple opponents. Another wound to the pelvis seemed to have been inflicted after death, a likely act of humiliation as his body was carried from the battlefield. These details matched written accounts of his final charge, his bravery in battle, and the mistreatment of his body afterward. But while much of the evidence confirmed Richard's identity, the Y chromosome results raised questions that were far harder to answer. When scientists compared Richard's Y chromosome to those of living male descendants from the Plantagenet line, they discovered a complete mismatch. Richard's DNA belonged to a haplogroup rarely found in Britain, while the Somerset descendants belonged to a different haplogroup, one more common in Western Europe. The difference was so large that it could not be dismissed as a testing error. It revealed a clear break in the male bloodline, a sign of a false paternity event. This meant that somewhere in the family's history, a child had been fathered by someone outside of the official royal line.